Yeah, kind of going back to those, the early days, the methods of computational neuroscience, the idea was to try to implement principles of biological intelligence in synthetic devices, sort of the early days, trying to understand learning from a computational perspective. And that really has continued. So, this course, embraces a similar theme. And so one of the things I like to do in talking about the hippocampus, and my training is as an engineer, electrical engineer, is to try to give intuitions into what I refer to as the computational imperative. Why do you need a structure like the hippocampus? The hippocampus, as I'll show here, evolutionarily conserved structure in the medial here portion of the temporal lobe. So here you see this kind of cross-sectional, the overlying neocortex removed. You see the hippocampus, which lies sort of in the middle of the brain. And in cross-section, you see here, this is a slice through it. You see the very simple architecture referred to as this is the trisynaptic loop. Information comes in from across the brain, converges on the adjacent entorhinal cortex, and then from the entorhinal cortex, it then goes through 3 synaptic jumps along this pathway, the perforant path providing the primary input, perforant path to cells in the dentate gyrus to CA3, CA3 to CA1, CA1 subiculum and out, CA1 also has direct projection. So it's this hop, 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 and then back out. And this architecture, you've had lectures talking about, visual cortex, this, structures in the neocortex have the so-called isocortical architecture, 6 layers, a lot of, sort of complexity, heterogeneity of cell types. While this structure is a much simpler archicortical organization, 3 layers. So you've got all these cells, sort of these kind of cells lined up here, and then apical dendrites, and then output. So it's a 3-layered structure instead of a 6-layered structure, very simple. The three different hops go through 3 structures that carry very distinct architectural features. The dentate gyrus, this is one of the structures, along with the olfactory bulb, in which cells, the principal cell, the granule cells here, undergo continued adult neurogenesis. The cells continue to divide and multiply. So your dentate gyrus, one of the only structures in the brain that continues to grow. So after your partying, you lost probably, who knows, 10, 20% of your brain mass last night. Well, dentate gyrus continues to grow and continues to add cells. And so it's sort of interesting, there are about 10 times as many dentate gyrus cells as there are pyramidal cells or principal cells in the other region. So it's kind of a unique structure. Lots of cells that continue to grow over the lifetime of the animal. These CA regions, CA standing for cornu ammonis or Ammon's horn, that's the structure of because it looks like a ram's horn when you actually dissect it in the human brain. These form the principal processing and output stations. And CA3 and CA1, the cell types are virtually identical, very similar pyramidal cells, but the connectivity is very different. CA3 has this dense kind of auto-associative connectivity. About 3 to 5% of the cells are connected to one another. In other words, I record a cell, and then 3 to 5% of all the other cells in CA3 are going to have connections. So there's like this very dense recurrent structure. CA1, same kind of cell types, but the complete opposite. Virtually no recurrent connectivity. It's a feed forward. So here you have this kind of feedback auto-associative network, feed forward network. And so you kind of have these architectural features, and these structures have remained evolutionarily conserved. There have been some variations. So for instance, you could say, those, the neuroanatomists, they're obviously not mathematicians because they forgot and they went from 3 to 1. What happened to 2? Okay, there actually is a CA4, sort of an intermediate region here, and a CA2. Now the region you don't see CA2 illustrated on here is because when you look in the rodent brain, CA2 is just very thin. It's a very thin region, doesn't make up a lot of the brain volume. But when you look in the human, and so evolutionarily, what has changed is CA2 has expanded massively. relative to those other structures. And you say, okay, so CA2, what's the deal with CA2? Is it, well, kind of CA2 is somewhere between CA3 and CA1 in terms of its architecture. One of the interesting features is that CA3, it's got like this super dense autoassociative connectivity, but it also gets this input, as I said, this trisynaptic input from the dentate gyrus, these so-called mossy fibers. Granule cells project in, they make synapses right near what's referred to the proximal apical dendrite. So you got the cell body and then you got dendrites that bring in information. Well, these mossy fibers, they make connections right onto the base of the dendrite. And they call them mossy because they make these huge presynaptic terminals. So huge that a single action potential is capable of driving the output of a single cell. So normally you would have like a pyramidal cell, typical pyramidal cell, couple of hundred inputs required. You kind of integrate maybe 100 inputs to get an output. But this input, one is enough. And so the idea, they sort of refer to these as detonator synapses, like one spike, one output. This allows dentate granule cells to essentially dictate the pattern of CA3. Now you think, okay, so if dentate can drive CA3, well, what's all this other stuff going on in CA3 going to do? Dentate's just sort of telling you, what to do. It's kind of like, I like to sort of liken this to, this is like in a classroom. This is like the instructor. Instructor is like speaking and driving the output. These are the students, and the students have the ability to kind of talk with one another. If you have the ability to exchange information, you can talk, that Wilson, what did he say? Oh, he talked about dentate. Well, let me tell you, I know something. Anyway, there's a conversation going on, right? Information shared among the participants here, but then you have this instructor that it can essentially dictate the output. And that is, I can ask you a question. I can ask you, this is the information I want you to give up right now, right? I can tell you what it is you're supposed to say and what you're supposed to think. Okay, but what about all of the conversation, the audio-associative interactions? Well, this is a long-winded way of saying CA2 has all of the recurrent connectivity, but the thing it's missing is it doesn't have the dentate input. So it's like an autoassociative network that is not under the direct control of this dentate structure, which, I don't know, it's kind of interesting. I liken it to, this is sort of like modern day instruction, where I'm up here talking, you have some ability to maybe quietly talk among yourself, but then you also have this very powerful external source of social communication. Like maybe you got your cell phones, you can say, oh, look, Wilson said dentate, let me check that out, right? It's like Google, right? So I have the ability to tap into a vast set of independent data that might be initially triggered or instructed by this dentate input, but then can be propagated through this expanded autoassociative or recurrent network. So you kind of think about, well, okay, so with human evolution, this kind of internal autoassociative component has expanded. And one of the things that people have looked at, and they've found that CA2 in particular seems to be involved in social cognition, social interaction. A lot of social cognition, quite frankly, is trying to initially interrogate, but then infer meaning from, sort of hidden content. I'm trying to understand, well, what did you really mean? Why did you, yes?
Is that social cognition independent of like auditory processing? Like is it actually social cognition, or is it just like if you're interacting socially?
If you're interacting socially, and so that the area is active during active social interactions, damage to CA2 impairs social interactions. Now, you can say, well, why are social interactions impaired? In particular in the rodent, as you point out, there's a lot of sensory-driven interaction, a lot of it auditory, but also a lot of it olfactory. And so you kind of think social cognition can be triggered by sensory input, but ultimately the question of now what do I do? I know what I see, but what do I do? And that is kind of guiding or sculpting responses is going to be due to, if it goes to the hippocampus, might be the result of this CA2 dependent processing. And the question is, well, what do you do? What do you need to do in CA2. What do you need the hippocampus for? Again, we get back to this computational imperative. What do you need the hippocampus for to do all of this? If I hear a sound, so I hear a hawk, I run for the bushes. Okay, that's pretty straightforward, right? I sense a female, I do approach behavior. I sense a male, I go and fight. Okay, so these are these kind of stimulus response driven behaviors. You don't need the hippocampus for any of that. You don't need the hippocampus for a lot of things. What you do need the hippocampus for are behaviors that involve an evaluation of what's referred to as context. And that is sort of the non-immediate cues. This could be things that happened in the recent past. This could be extrapolation of what might happen in the future. And then the identification of things within the environment, contemporary cues that are not directly tied to the response that might impact it. And so a good example of context might be, well, what do you think, what's an example of context that you could think of, like in modulating a response? As long as we go through every single day or night. It's like time of day. Time of day is a great example, right? You could be in here, same room, morning, evening. Now, while you're in the same room, what you expect and what you do may differ. Maybe at night, you come in here and it's like party time, right? You come in the morning, you have your coffee, it's lecture time. And so you learn to associate different expectations and then consequently different actions with these additional features, time of day just being one of those things. And so what you would want is, I'm getting exactly the same input, but I need to map it to a different output. The hippocampus allows you to represent that context. And what we'll see, so context is it's kind of a vague notion. It's any of those things that you need to take into account when you're mapping a fixed input onto a different output. And so
going back to the anatomical information you were talking about, I was wondering how those information were gathered and what the resolution is in terms of like the pyramidal neurons and the multifibers. Are there other cell types and how are their connections?
In the dentate gyrus. So there certainly are other cell types, and I kind of gloss over the complexity of the organization, even though the cell types themselves are generally, the same. They have a sort of a pyramidal morphology. When you look at the composition of the channels and the inputs, they look very similar. Of course, there is some heterogeneity, and they're kind of a mapping. And part of that mapping comes from, so here you've got this kind of long structure again, and this is like a cross section through it. Well, the hippocampus gets input from the adjacent, so right, kind of adjacent to it here is the entorhinal cortex. That would be adjacent to it. Entorhinal cortex sends in these inputs along the perforant path, and those inputs come in and they kind of run along the so-called sort of the transverse direction. But there's also longitudinal connectivity and organization. And so everything I'm going to talk about comes from a region in hippocampus. In the rodent, it's referred to as the dorsal hippocampus. That's the hippocampus on top here. It kind of curves down, and then down here is what's referred to as the ventral hippocampus. Now, while the dorsal hippocampus and the ventral hippocampus basically have the same kind of architecture, a little bit of difference, the relative proportion of CA3 and CA1 kind of differs a little bit, but the primary difference between dorsal and ventral is where does it get its inputs from. They all get their inputs from the entorhinal cortex, but the entorhinal cortex gets its input from the rest of the brain, and the entorhinal cortex is organized in this sort of kind of roughly topographic fashion, in which different parts of the entorhinal cortex get input from different parts of the brain. And in particular, the medial entorhinal cortex would project into the dorsal hippocampus. You don't need to remember all the medial, dorsal. It's just, it gets information from what are referred to as the spatial regions of the rest of the brain. And so when you think about processing, there are these processing streams. I like start from the visual, like sort of visual processing in the back. We go from the visual cortex, And then as it moves forward, the visual stream kind of splits into two paths, the dorsal and the ventral stream, right? You've heard of all this, like dorsal stream, you get like sort of motion information, shape, form, where are things. The ventral stream along the temporal lobe that comes along the temporal lobe here, that carries information about like object identity, so-called what information. So you have like the what and where streams, that's like, you know, and so this dorsal stream, the where information, that tends to converge here in the dorsal hippocampus, in the medial entorhinal cortex, and then into the dorsal hippocampus. The lateral entorhinal cortex gets information from the what stream. And the way I like to remember that, I think lateral entorhinal cortex also gets direct input from the olfactory system, the olfactory cortex, and even the olfactory bulb, which is kind of like the retina of the olfactory system. Direct input from the olfactory retina directly into the lateral edge of the entorhinal cortex. That's really what information. You get very little where. There's very little where information in olfaction. It's like, I smell something, where did I come from? Well, you know, maybe how would I figure out where it was coming from? If I had olfaction, I would have to move, right? I smell, oh, I think it's getting stronger over here. So I would have to couple that with some spatial, like some, you know, another system, movement system to figure out location. So olfaction is pretty much just object identity. That pretty much just projects into the lateral entorhinal cortex. And that tends to project into the more ventral regions of the hippocampus. So you can think of the entorhinal cortex, or let's say the sort of neocortical areas have kind of this separation of what and where. You got the dorsal where, you know, ventral what. In the entorhinal cortex that projects into it, you've got this medial where, lateral what. And then into the hippocampus, you've got the dorsal where, ventral what. Now, again, this is sort of broad strokes, but it points to the preservation of this relatively low dimensional component of representations across the brain, regardless of, you know, what particular modality, and that is the idea that you can think of our representations of information in the world as carrying these two primary dimensions of location and identity. What and where is something that doesn't get lost topographically. It gets conserved in some way. And you think, well, why would it do that? In particular in the hippocampus, where, as I pointed to, you have this sort of, there's like this contextual, this idea of context. What does that mean? The sort of the context as defined by location, where, and context as defined by what? And that is, and I'll sort of give you another little clue here. Ventral hippocampus also strongly implicated in social kind of, you know, social learning and memory. Dorsal hippocampus more in spatial. So you have, if I damage the dorsal hippocampus, man, I can't find my way around. I can't, you know, where did I put my keys? I can't find my car. I damage the ventral hippocampus, I'm pretty much okay with that. Vice versa, if I damage the ventral hippocampus, I find deficits. I can find my car, I can go to my, I can find my way to the lecture hall, but I have deficits in my social cognition and social contextual awareness. And so social interactions affected here, spatial interactions affected here. And another clue is when you look at the output, primary output from the CA and subicular regions of the dorsal versus the ventral. Ventral is more prefrontal and amygdalar. The dorsal goes to sort of a region, the kind of the spatial regions of the parietal cortex, in particular the retrosplenial cortex. Anybody talk about retrosplenial cortex? Retrosplenial cortex used to be one of those, it's like, and Adam's always making up names for stuff. But, you know, retrosplenial cortex is sort of thought of as like a hub structure in the so-called sort of the default mode network. This kind of resting state, you know, area that's active when you're not doing anything. So it's like, oh, if I'm just kind of sitting quietly thinking. this sort of default mode network gets activated, and retrosplenial cortex is a part of that. It's also kind of a hub. It's sort of a link between, as you move from, like in the visual, the dorsal pathway, basic visual information, higher level visual associational information, and then you get to retrosplenial, and then you get cingulate cortex, prefrontal cortex. So it's kind of between sensory and then prefrontal processing, the decision making. You record in there, and either in like human fMRI or you do electrophysiology in rodents, what you find is it can be involved in the transformation between egocentric to what's referred to as allocentric. And so we'll talk a lot about allocentric. Allocentric is like things out in the world. So as I'm moving around in space, and I kind of see you, and as you move, and as I turn my head, okay, like Chris is over here, he's over on the right, he's down low, okay. So I have areas, the visual cortex is going to be very good at keeping track of where is Chris in my visual field. But as I move, okay, now he's on the left, left visual field, now he's up. So where he is relative to me as I move changes all the time. Where Chris is in the world doesn't change at all. In fact, it hasn't changed in it's almost 10 years. Chris has been sitting there, right? So his position in the world remains the same. So somewhere I have to transform stuff that's changing in the world because I'm moving. And then I have to map that into things that are changing in the world independent of my movement. So you go from like me, egocentric, to world, allocentric. And what we'll see is that, so that structure, the retrosplenial cortex, is at that nexus where it's transforming, kind of bringing together the egocentric and the allocentric. So okay, sort of, why is that important? And that's the region that's directly interacting here with the dorsal hippocampus. So why is that important? Again, this sort of contextual, thinking about like contextual processing. When you think about like context-dependent responses, context-dependent responses are context inappropriate responses. So a good friend of mine, Lamellar, who studies the prefrontal cortex, likes to use this example of sort of context-dependent processing, like sensor response gating. So when you hear a phone ringing, your natural response, you hear a phone, you like answer the phone. That's just what you do. You hear a phone, you answer it. But now suppose you go over to your friend's house and you hear your friend's phone ringing. Do you answer it? Okay, in the one case, if it's your phone and you answer it, that's the correct thing to do. If you're at your friend's house and you answer their phone, it's like, whoa, dude, what's going on? And you don't answer somebody else's phone, right? That's just not what you do. And so the context makes a huge difference. Exactly the same stimulus, exactly the same response, but you have to gate it. One is appropriate, one's inappropriate. A damaged prefrontal cortex, it's like, okay, if somebody is answering your phone, probable, like some kind of, sort of frontal disorder, some problem with frontal processing. But now you could also have the same kind of damage or the kind of inappropriate response can occur with damage to the hippocampus. And that can come from, it's like, oh, I know that I shouldn't answer, my friend's phone, but I was not able to figure out, am I at my house or my friend's house? I'm not really sure. In other words, you can a deficit in context recognition can be very similar to a deficit in context, loss of context-dependent response gating. The two structures, prefrontal cortex, hippocampus, kind of working hand in hand. So that's just like this, okay, that's your house, my house. Now, if I record in the hippocampus, what I would find is that cells in the hippocampus, the representation of context changes for the two. That's how you can tell the difference. There's a so-called map, a different map for the two different contexts. Anything that refers to like a change in context that should be associated with a change in response could be like, morning and afternoon. You say, okay, morning and afternoon, different context. Should I have a difference in the hippocampal representation? And the answer should be, well, yes. And when you actually do record in the hippocampus, under conditions where something like time of day, no other cue has actually changed, it's only a difference in time of day, that's sufficient to change the representation of the map. People refer to that change in map as remapping. So you can trigger remapping, change in the pattern of activity of the primary cells in the hippocampus as a consequence of change in anything in the world that should dictate now a new kind of response mapping. So anything where I have to pay attention, geez, you know, it seems like it's the same, but I need to do something different in order to make it appropriate. And those subtle, and those changes, they could be large. It could be like from your house to your friend's house, right? Okay, that's probably pretty different. Or they could be very subtle. Exactly the same room here, nothing has changed, just time of day. And so the idea that the nature of the remapping needs to be triggered under conditions that could come from lots of change in the environment or few changes in the environment. So this is the very, very long-winded way of getting back to this question about the dentate gyrus and what might be the role of the dentate gyrus. So the idea with the dentate gyrus is you have this vast expansion of potential representation space, lots of cells, about 10 times as many. Estimates are about 1 million dentate granule cells in the rodent, relative to about 100,000, 1 to 200,000 pyramidal cells. And the firing of dentate granule cells is very sparse, and that is they tend to fire at low rate. It's hard to get them to, to fire. But that they become active under conditions, you can get them to remap under conditions of novelty. So when things change in the environment, dentate granule cells can respond to that change. And so the idea is that small changes in the input can lead to large changes in the output because you have this kind of sparse distributed memory. Sort of think of like, computationally, sort of like a hash function, a hash table, right? Hash table, you sort of take an input and you map it onto something completely different. It doesn't really make any difference what the input is. It's just if it's slightly different, you just map it onto a completely different output. And so it's the idea that you kind of separate, you sort of hash inputs independent of their similarity. And so this idea of pattern separation is something that has been embraced as a kind of a computational principle that the dentate gyrus could implement. But it's this idea, the general, whether you do it through, through like sort of hashing, through sparse distributed representation, it's the idea that that you need the hippocampus to be capable of remapping or changing its representation in a way that's not strictly dependent on the distance between like the input vector. That is, yeah.
Do you think that that's true across species? So as you go, because there's kind of this discussion about whether or not there's pattern separation in the human hippocampus or not.
Great, it's a great question. So thinking about that, oh yeah, sort of pattern separation, is that something that's been conserved? And when you look comparatively, evolutionarily, the biggest differences that you find, there's like the CA, kind of the CA regional difference, but the dentate gyrus and the relative proportion of the dentate gyrus changes dramatically. And so like sensitivity to the kind of the remapping capacity, pattern separation capacity does seem to be quite different across species. Now, again, thinking of that strictly as pattern separation, I don't think is exactly the right, it's not exactly the right way to think about it, but the dentate gyrus, and again, then you kind of think about, well, why do you have this continued neurogenesis? Why do you continue to need new dentate granule cells if all you're doing is just this pattern separation and through kind of sparse representation? Why does that necessitate the addition of new granule cells? There was another question related. So there's no good answer to that. I mean, there are answers there are answers that I could give. And they would have to do, there's the kind of the mapping function, all these things that we're talking about, mapping functions, none of them have it, I haven't even used the word memory yet. So the idea of memory, and that is how do we take in, retain information, and then use that information subsequently. So there's the simple model, the storage and retrieval model. Sometimes it's kind of used interchangeably with this the idea of engrams or memory engrams. And there's like a pattern, this is sort of the photographic scrapbook idea. I have an experience, there's some sort of high dimensional representation that has all the details. Somehow I have to capture that, I have to store that, I have to be able to invert that so I can go back to the details, and so I have a memory of this particular event. Maybe I have a memory of this particular lecture, and I have to be able to separate that from other similar lectures that I've given. And so I have to be able to timestamp it. I have to be able to retrieve the constituent elements. So that's the storage and retrieval model. And so a lot of that has kind of the idea that the dentate needs to contribute in some way. I need to separate different patterns for different memories. But then there's another model. The other model is the kind of synthetic generative model. And that's the model I favor. And the simple version of that is all memory is false memory. And that is that all memories, when we retrieve them, they are actually synthesized or generated from kind of a constitutive model that has been built by identifying statistical regularities in past experience. There are things that differ, there are things that are similar, and so what I need to do is I need to find a lower dimensional representation that captures those things, and then when I retrieve, I'm essentially, I'm kind of generating, something that is consistent with, but not, kind of vertically accurate relative to a prior memory. And so then the idea is, how do I build a model? How do I incorporate information? How do I do kind of structured dimensional reduction that will allow me to do this kind of appropriate context-dependent retrieval? And so that's the idea, and that is that things are going to change, but what I'm really looking for are those things which are conserved and consistent. It also relates to an idea, and Boris, we've kind of talked about this in the past. It's this idea of breaking down representations into reusable components, this idea of compositionality, and that is that kind of an effective strategy for representations, rather than just sort of randomly storing everything and retrieving it, very inefficient, rather than just doing kind of arbitrary, compression and dimensional reduction, which can come through. This is like kind of sort of standard, kind of deep learning. You just sort of dump in lots of data. It will actually do, kind of some sort of kind of structured reconfiguration of representation to capture shared components, a lot of the things that I actually described, but it won't necessarily do it in a way that can anticipate future, sort of future needs. And so this is like what we often refer to in sort of the origin of CBMM is the contrast between biological learning and machine learning is that, sort of machine learning, the performance improves as n number of samples in the training set goes to infinity, right? That's the idea, big data. You just train with more and more and you're able to, kind of identify these effective representations. Biological intelligence, there you have to perform, the idea is that it performs well as n goes to 1. And you can even extend that, you could think as n goes to 0, and that is I need to have kind of a built-in, a model that pre-exists that can anticipate things that doesn't need to be trained on, a large number of exemplars. A good example of that would be obviously the kind of stimulus response learning that we already talked about. I see an owl, I run for the bushes. Okay, I don't want to have to train that on many, many examples because I'm not going to get many, many examples, right? In fact, I might not even get one example. That's an example of, I need to perform as number of training, samples goes to 0. I need to already have that built into the model. And so the idea is, can I identify regularities in the data drawn from experience that will be able to predict and anticipate future uses? That's this idea that if there's some intrinsic compositionality, a way of breaking up things in a way that will capture future regularities, that's in a way better, particularly in a real world biological context, than just extracting regularity through large amounts of data. Extracting through large amounts of data is great if every case, right, I know nothing about the data, right? I know nothing about it, obviously, okay, I just want to train on it. If I know something about it, if I've trained previously on it, well, I would like to use some elements of that previous training right? Not start from just like a, sort of a randomly connected network.
I feel like there's still some similarity in the sense that you can kind of argue that the brain is built through evolution, so that evolution saw lots of cases. And then you can kind of also use the predictive kind of framework in the artificial neural network, where you say that like the way that it is kind of like a trained model would also identify the useful regularities in the data and draw from the experience that is already in the weights.
Already in the weights. So I need something new, right? Train on new, but I need to conserve the old. And so it's like I don't want to. If I have one network and I just continue to to train it, to incorporate it, and everything changes, it's the most flexible, but it's also the most inefficient. Better would be, and then on the other side, it's like I have a pre-wired network, right? The weights are already built in to kind of anticipate, and I just make subtle changes, right? I make subtle changes to adapt to, let's say, the particularities of the new input. But then somewhere in between there's the stuff that's old and works, and then there's the stuff that's new. It's like something I see that I've never seen before. What do? Well, maybe I need a new network for that, right? Maybe I need a new network that I can kind of train up and integrate with my older network. So you can think of having, well, not like one network, the hippocampus, where you just train everything all the time on new inputs. What if you had like multiple networks, many networks, all being trained simultaneously, and then you could control the interaction between them? Well, I kind of left out one kind of, historically, this was something that had been pointed out anatomically about, in fact, it's actually, I don't know if they mentioned it, Emerald and Witter, it's referred to as the Lamellar hypothesis. One feature of these hippocampal projections is that the input from the dentate gyrus to CA3 here, these inputs don't go everywhere along the hippocampus. In fact, these inputs on the dentate gyrus are actually, they're confined to a narrow strip or a lamella. So you can think of like CA3. CA3 connects both sort of transversely, but also longitudinally. The dentate inputs are transverse inputs. They lay out like little slices. So it's like the dentate gyrus not only can select which CA3 cells are active, the pattern of CA3 cells, but their activity can select the lamellae, which of these kind of slices of the hippocampus. So now you're thinking, oh, wait a minute. So it's like, so there's this organization. It's not like just sort of one, three-layered network. You've got all these like sections. They can all be selected or gated through the dentate gyrus, and then you've got the ability to do auto-associative interactions between them, and then they project out through CA1. So this idea that there's this additional lamellar organization suggests, and this was originally when it was observed, the thinking was, well, maybe you have like this parallel process in the hippocampus. It's not just one, sort of massive network. It's a whole bunch of parallel networks that can communicate with one another. And so when you think about this, and so I think that I kind of threw out the idea, well, this idea of compositionality, the idea that you kind of break up things into usable pieces that can be reconfigured, right? You just reuse them in a flexible way, and that kind of accelerate learning by taking advantage of previous structure. And so the idea that compositionality might be something that's embedded in this lamellar architecture. Yeah.
Earlier in the talk, you introduced the presence of neurogenesis, but the context that you introduced it in was primarily for restorative. Sounded like it was more of a restorative capacity for like the loss of neuronal connectivity, but here it also sounds like there might be an additive.
It's additive. So actually, dentate volume increases. So over time, you continue to add new capacity. And so again, you kind of think of what would that added capacity, what would that look like? What would that actually translate into? And how might that be applied to modulate the kind of the lamellar selection? And so the idea that, well, what does it mean to have like sort of modular or compositional organization. So you have representations. So you think of each of these lamellae as being like, I don't know, it's like, in the simpler view, oh, it's like one context-dependent slab, right? I've got one slab for, I don't know, for, Woods Hole, another slab for, Boston. I mean, it's not like that, but, the idea is that you have some ability to map these lamellae into, like, reusable components. So that I could have the maps for Boston, if they were all in one lamellae, okay, great. If I had Woods Hole, if I had all the separate maps there, that would be great for like pattern separation. No interaction between two. Anything I learned in Woods Hole would explicitly not apply to Boston. But suppose there's stuff that actually does apply between the two. Maybe there are things that I experienced in both. So maybe I would like to have some way of like training both Boston and Woods Hole with this new input. Now, how could I do that? So that would be potentially one of the functions through this, kind of dentate lamellar biasing. I could kind of select the context-dependent components that would then be trained in order to kind of establish this kind of structured, shared, potentially compositional-like organization. So I could separate the things that are different, I could combine the things that are the same. And so I could modulate in a way, the way in which I integrate information through this kind of selective gating. It's very much like I tell students whenever I give lectures, and everybody comes in the first day of lecture, I say, I don't know exactly how all of you are going to perform, I don't know a lot of it, but there's one thing that I can predict about each and every one of you, and that is when you come back into class tomorrow, you're going to sit in exactly the same spot. It's just one of these things. Everybody comes back and you tend to sit in the same spot. It's like, why do you do that? Well, it turns out that the cells, as maybe we'll get to, the cells in the hippocampus, referred to as place cells, they respond based on location. And so the collection of hippocampal cells that are going to be used for the encoding and processing of all the lecture material you're going to get kind of depend on where you're actually sitting. If you sit in the same place, it's like I'm training the same set of cells. If I sit in a different place, I'm like training a different set of cells. And so I say you can actually use that if you want to like integrate across lectures, you sit in the same place. If you want to separate across lectures, like, man, I got two lectures in here. I got one on the hippocampus, and I got, one on music theory. I don't think those are related. I think I'm going to sit in a different place in the music theory. And so that would be like what the dentate could be doing, saying, no, let's not get the Boston Woods Hole, let's not get the music theory and hippocampus lecture separated. So we'll just we can kind of select which maps get selected. Yeah, question.
Is there good evidence of place cells in people?
Good evidence? There certainly is evidence. And we'll kind of get into, what constitutes like sort of a sufficient demonstration of place firing. And as I'll talk about, the most definitive is the direct measure of action potential output, putting in electrodes, recording spiking activity. Indirect measures would be like metabolic measures. Functional MRI could be other, ECOG or other methods for sort of looking at sort of proxies for spiking output. And so I would say, the equivalent has the kind of the direct equivalent of place recording electrophysiologically, hippocampal cells and people that are moving around. Has that been demonstrated? No. Have there been direct recordings in the hippocampus of people? Yes. There's all this work, particularly with epileptics, where they can record, and you find that you get, hippocampal responses to, complex cues. You get spatial responses in virtual environments. We'll talk about like virtual environments and real environments. One of the things that kind of separates the virtual, from the real are the nature of the motion signals. And we'll kind of point to movement as being something that's really central in the rodent hippocampal representations, and not just place cells, but there are other properties of place cells, in particular the preservation of information about time or spatial sequencing. So the hippocampus actually represents not just location, but sequence of location, so-called trajectories, and that is what's the recent past, what's the recent future, and that's really kind of core to the nature of hippocampal representations. And so this idea of, we'll go to slide number 2. Spatial and temporal processing is central. Yeah, go ahead. Question? No? That's you? Yeah.
Yes, I was implicitly, but I was going to raise my hand, yeah. It's just, so about sort of spatially related maps, I guess we can call them. To what granularity do you think that they exist?
Yeah, great question. See, so now I couldn't, and I got to go back to them. So one thing, so when you look at place cells, and this is some of the difficulty when we talk about are there equivalent place cells in humans?
But we navigate quite differently than rodents. Like we do mostly landmark navigation in a different way, right? Because we can have a top-down view of something.
We do, so there is, so there's like, yes, sort of landmark-like navigation. So it's kind of interesting thinking about like the egocentric, the allocentric, landmark navigation, which is, it's the kind of thing that you see in retrosplenial cortex, for instance, like this sort of top-down navigational strategy is something that is impacted. You see in the retrosplenial cortex, and in the hippocampus, it might make use of that. You say, well, this kind of map-like, organization, does the hippocampus generate that through its place cells, or is it actually given that through its input? And I didn't mention this, but some of you might be, sort of familiar with this. Both place cells and its complementary input cells in the entorhinal cortex, the so-called grid cells, were given the Nobel Prize, right? This is, how long ago is it? Almost 10 years now. So, and the idea with the grid cells is there's this kind of map-like metric structure, right? So you can take the idea of a topographic organization, a map in which you can you can see the relationship of everything, sort of the distance and location representation of everything. That's present in the entorhinal cortex, and in particular, the regions of the entorhinal cortex, the medial entorhinal cortex that project into the hippocampus. So back to this kind of granularity question. So the place cells, as they originally identified by John O'Keefe in 1970, were these cells that have, I'll show you an example of them, have these tight little place fields. They fire in this location, but not in this location. So you really get you think, oh, this could really map out locations in space that I could use to navigate if I wanted to figure out, where I was. So you kind of look at behaviorally, what's the spatial navigational resolution of a rodent, and it's about 2 centimeters. And so this has been tested. You put a kind of a food grid. It's like a little grid with little holes in it, and you ask, okay, how fine a discrimination can they make between 2 adjacent holes? Food's in this one, not in that one. And it's about 2 centimeters. So you can say, okay, that's the resolution of their representation. And that's consistent with the resolution of representation that you find or estimates that you find in the dorsal hippocampus, the finest resolutions. Yeah, okay, if I decode dorsal CA1 hippocampal activity, I can kind of get it down to about that same scale. As you go to ventral hippocampus, as you go along the long axis, fields get larger. You start to see different, sort of different properties. They're like symmetry and regularities that pop up. And it suggests, maybe it's not strictly about, sort of precision localization. Really, maybe it's more about providing a broad spatial context to the kinds of signals, the non-spatial-like signals, like the social signals, the olfactory signals that come in. So you provide a general social context. It's like, yeah, this place, that's the smelly room, and that's not the smelly room. It's not like, oh yeah, this is the smelly location, and 2 centimeters, not smelly location, right? So the resolution required to provide that kind of, it could be the same thing. It's like the nature of the social interactions, pretty much the same if I'm over here as they are when I'm over here. Now, if I'm actually plotting I'm trying to navigate, it's like I want to figure out what's the fastest way to get out of here, but, if there's a fire alarm, right? That kind of planning, I need to be able to think about specific paths, what's the distance, how long would it take? That's like a dorsal CA1 function. So you can think the resolution, small place cells, high degree when you decode, very precise localization, consistent with the precision of localization that you can achieve behaviorally. Ventral hippocampus, big place fields, they're not linked to navigation, but presumably, it might be related both to the inputs and to the nature of the representation. And so when I kind of get to this, the nature of the representation. So we talked a lot about like space, spatial context. But the other thing I pointed out, and that is that it's not just the cues that are present now. It might be like cues that were present in the past and things I anticipate in the future. When we think about memory, that's sort of what we think about. And that form of memory, we generally refer to as episodic memory. In particular, episodic memory being that autobiographical, and that is memory about things that happened to me that carry this kind of unique time signature, that is, unique sequences of events that can be recalled. So that's what makes up an episodic memory. Linkage of events, context-dependent, autobiographical. Navigation, linkage of spatial locations, and both are impacted by hippocampal lesions. So in humans, you damage the hippocampus, navigational deficits, but most profound, or most obvious, I would say, are the episodic deficits. So that's what got the hippocampus all the attention in the 1950s, the case Henry Meliasen, famous case, he was referred to as HM before his passing, underwent this surgical procedure involving bilateral resection of medial temporal lobes to treat epilepsy. Hippocampus, because of its architecture, often is a site of seizure initiation. So they took out portions of the medial temporal lobe bilaterally, and subsequent to that, he lost the ability to form any new memories of his experience, also impaired in sort of navigation. But the idea that the hippocampus, one structure, damage it, you can't form any new memories. And there was sort of an interesting kind of twist to that, and that is, following the surgery, there was some retrograde amnesia, and that is loss of recent past events, but there seemed to be some preservation of so-called remote memory. You ask him things about his childhood, he could tell you stuff that seemed to suggest he could remember things from the distant past, even though he couldn't form any new memories of the future, and he didn't remember recent memories. So there was this idea of kind of graded retrograde memory consolidation, that this is a slow process. Hippocampus stores new information, and then it slowly transfers those memories elsewhere in the brain, so-called systems consolidation model. And that other site might be somewhere in the neocortex. Now, there's nothing that suggests explicitly that actually occurs. Again, this is sort of like the engram model of storage and retrieval. I store, right, I've got like my scrapbook, I store my memories, and then over time I move my scrapbook from my bedroom up to my attic, and it's just like this slow process. Another version is that, again, it's sort of this synthetic generative idea, and that is that there's some recent, the hippocampus is involved in proximal storage experience that I have. Maybe I take it in, I remap it, I encode it in some way, and then there's a gradual process by which I train a separate network to incorporate that information, and that then the subsequent memories, so-called remote memories, when I ask HM, do you remember your childhood? It's false memory. Again, he doesn't really remember his childhood, but he can retrieve things that sound like, seem like they are consistent with such memories. And so now you kind of think, fast forward to today, you've got all these like the transformer models, all the, like ChatGPT, where they sort of work in the same way, right? They don't really, they've been trained on a lot of information, but you never really get back any of that information. You get back stuff that is related to that, and the so-called like they hallucinate. Well, so you could say, yeah, HM was like hallucinating, right? He had the benefit of gradual training of this other network. And so hippocampus is training the chatbot. It is not the chatbot. And so you kind of think about that process. Episodic memory. What is that? Why am I not able to form any new episodic memories? So I'm going to kind of ignore for the moment this kind of past, the loss of access to recent past memory and the apparent preservation of remote memory. I'm just going to throw the remote memory into, that could be like another process, right? It's generative, it's not really, it's just a separate thing. But the inability to form any new memories, and that is I can't remember anything new if I damage the hippocampus, either navigationally or episodically. And so the link between those two things, I would argue, because you can have plenty of episodic memory that does not require that I actually, let's say, necessarily have a spatial element to it. I could be watching a movie, for instance, and it's not, I'm sitting in the same place. Stuff is changing, cues are changing, but my location, allocentric location, is not changing. And this is where you can get into the discussion about in humans, do you see place cells for things that are like, watching a movie? There can be responses, for instance, place-like responses based on eye gaze. As I navigate a visual scene, there can be place-like responses. So there are ways in which the hippocampus can remap changing inputs, either relative, like eye gaze dependent egocentric information, or like stationary temporal information, movie information. Gabrielle's done things looking in the hippocampus, people watching movies. You get the same kinds of responses. But one shared element of both of those is that they represent kind of unique linkages over time, and that is the sequential dependence of the activity is something that needs to be conserved. And so I sort of point to sort of modern transformer models. There are lots of things that go into these sort of transformer models, but one of the things that was that conferred a sort of a potent computational ability to accurately generate synthetic streams that that sort of seemed to be kind of appropriate given the training data was taking temporal information. So you take different time points and then you map them into different layers of the network. And so instead of just taking sort of identifying statistical regularities across different points of a static input, you can now identify regularities across inputs in time. So that's one of the things the hippocampus does. It can take things across time. So the idea that time sequence is something that can be important for identifying, separating, and representing inputs is also something that would be important in training this generative model, and that is that the hippocampus, it doesn't just need to to identify or recognize something if it was similar than before, but it does need to to work with, for instance, the prefrontal cortex to say, oh, the phone is ringing, it's my friend's house. Should I answer it? And the prefrontal cortex will generally say no, but it's the sequence of events. What just happened, what's happening now, and what do I do next? That's that's this component. You could give lots of examples. It's like maybe, the last time I was here, my friend, he couldn't answer the phone, so he asked me to do it. And so I might have this context dependent, I kind of string together past, present, and future in a unique way. And so we're going to be thinking about these two things, mapping, remapping, and then temporal sequence representation. And then the approach, I'll just quickly run through this, just direct electrophysiological recording, putting in electrodes, recording spiking activity. This is a little device. It's an electrode array. These are little micromanipulators. You can drive the electrodes down here. The dimensions here, this is about like 30 microns across. Individual cells are on the order of about 10 to 20 microns, maybe 10 to 30. So individual neurons are about on the scale of these individual tips. And so when you put these electrodes into the brain next to collections of cells, you can kind of triangulate the relative position of ensembles of nearby neurons using the kind of the spatially dependent attenuation. Cells that are closer to this electrode, action potentials, the spikes will be larger than they are if they're farther away. And so you can use this relative amplitude difference to separate or identify cells. So it's just a way I can put in a couple of wires, I can pull out bunches of cells, dozens of cells, let's say from an individual electrode. I put in dozens of these electrodes, and now I got like hundreds of neurons. So what do you do if you can record from 100 of individual neurons? And so this is a kind of an example. I'll just kind of run this, and you can kind of hear, this is like the raw data. So if you're sitting in a setup, this is the video screen, you see the animal, you would hear the spiking on your audio amplifier. You would map out each of these points as an identified action potential. This is the amplitude of the action potential on two of those wires. And so you can see, again, there's a separation, like the little blue cell here. It's large on this channel, small on this channel, which means it's closer to this wire than to this wire. The red one's in between. So these different cells form clusters based on their location in space. So now we can kind of map out where is the animal when these cells fire. And this is the space, this is the so-called place cell-like property, where the light blue cell fires here, the dark blue cell fires here. The animal's running. Now, another thing you can hear, to me, it's just like jumps right out. You might not be able to hear it. There's this rhythmic modulation, the so-called theta rhythm. It's a 10 hertz modulation. It's kind of which you heard very prominently as the animal actually ran across here. I'll do just one more time for you, so you can hear when the animal moves, you get this rhythmic modulation. When the animal stops, the rhythmic modulation goes away and you get this bursts of activity. So there are really two modes there's a little burst. So see, if I'm sitting here and I'm not even watching the animal and I hear that, I know, oh, the animal stopped, right? When I hear the rhythm, it's like, oh, I know the animal's running. That's stopped, okay. He's like sitting there, a little bit of movement. Now he's there, and I hear now he's like clipping along. Now he's running again. So I can hear all of this as just based on the background rhythm. And it turns out these two modes, which you can switch between very quickly. So here, I don't know if you can hear that. Right there? Okay. So you get a trained ear and you pick that stuff up. So there's two modes that can switch between very quickly, rhythmic, arrhythmic. And those two modes, it turns out, process information very differently. And one way to actually see that is if I now take the same activity, instead of showing you the spikes, I'm now going to use a simple sort of Bayesian decoding algorithm to estimate conditional probability that the animal is at a given location given the current pattern of spiking activity. So given that these cells are firing, what's the likelihood that the animal's in any given location on the maze? And I'm going to show that probability with the size of a triangle. The triangle is going to give, that's the sort of the Bayesian estimate of location. And so what you see here is if I do this, when I decode activity when the animal's running, the triangle tracks the animal's position. Place cells, place coding. And again, as I said, I can decode down to about 2 centimeters. When the animal stops, triangles go away, hippocampus no longer representing current location. What happens to those triangles? If you see, what they do is they very quickly, animal's going to stop, burst, jump around the maze. Okay, so you get the quick, right? There's many locations represented at remote locations. And so when he's running, you'll see these, tracks. And so you get these two modes, present state and then this remote state. And if you take the animal off completely, yeah, go ahead. Question, yeah.
Do the cells also track speed? Can you decode speed of motion as well?
Speed of motion, absolutely. You can decode speed of motion. In fact, as we'll kind of get to, yeah, we're almost at break time. We're going to get to another property of hippocampal firing, and that is there's this, that, not only is the firing rate modulated by the location of the animal, but the phase with respect to this rhythm, so-called phase coding or phase precession. The phase changes dependent on where the animal is relative within its place field. So if I actually look at the timing with respect to the rhythm, I can get a very precise localization. If I look at the firing rate, and you could kind of hear it as the animal ran faster, it's like there were more spikes. Firing rate tends to correlate with running speed. Firing phase tends to give the most precise spatial information. This led John O'Keefe to actually conclude when he discovered phase precession, the phase coding of the hippocampus. He concluded that actually the hippocampal code is, the spatial code is the phase code is the spatial code, and that firing rate is actually the velocity code. Yeah.
So is this a session after the animal gets enough exposure to the maze? How much learning does animal need to generate this kind of effect?
Great question. So the place cells. So this was the original, the original study when I started doing, developed these implants and was recording population activity to kind of answer that question, because it can be very hard if a cell only fires at one location. Well, how do you know, right? What's going on at all the other locations? If you record lots of cells, you can see all of them simultaneously. And what you see is that there's very rapid expression. One time through a location is sufficient to identify the location where that cell is going to fire. But with repeated passes, the place the place-dependent firing changes. You get more firing, it's more consistent. So you could say like the firing rate goes up, but it's actually more the consistency of firing increases. And in fact, when you look at it in more detail, what you find is there's like this novelty code. If I'm just looking at activity, I'm looking at a population, I can kind of tell you how familiar an animal is with a location or environment by just looking at the covariance across the population. If I get a high degree of consistency, like all the place cells are firing each and every theta cycle, so like this 10 hertz theta oscillation, it's kind of like a clock. It says, okay, you know, measure, 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 and if each time I measure, I get the same measurement, it's like, okay, I'm pretty sure this is the right location. So the consistency, as reflected in covariance, is correlated with familiarity. What happens in novel exposures, novelty, the cell is going to fire, there is a lower rate, but what that lower rate means is really, if I'm like sampling it 10 times a second, it's like, okay, this sample, yep, fired, next one, no, next one, oh, okay. And so if I get 10 samples, maybe I get like 3 or 4 of them, and like 3 or 4 for this cell, I get another 3 or 4 for other cells, the probability that they're all firing in an individual cycle goes way down. So I can tell, is this the first pass, is the third pass? What happens is the consistency across the covariance increases, and I can tell you the relative familiarity based on that. That also translates into decodability. So the decoding, particularly if I'm using like a simple, you know, sort of a, you know, probabilistic decoder, like a Bayesian decoder, accuracy of decoding will increase as covariance or consistency goes up. So you can think about that as like, yeah, consistency, covariance, accuracy, those are the things that change. But the first pass is enough to kind of identify which are the cells that are going to participate, and then over time, how they participate is what changes. Yeah?
Do you have an idea for the spatial resolution that you need to have for the scanning modality to actually do this decoding test successfully? Because it had to be like single cell resolution or...
Well, not, when you say to do the task, so there's the decoding, what I have to do, what the rat has to do. So, yeah, the decoding, so this was, again, so this original paper I did was population, you know, population recording. I was actually looking at decoding accuracy, and the decoding accuracy kind of asymptoted at about 100 cells. So, you know, after about 100 cells, I had enough information to decode location. Now, so that then begs the question, okay, so 100 cells, but if you look at the relative sparsity of, you know, of a representation, you have about 1 to 5% of hippocampal cells active at any given location. And so if you take that, like 100,000 cells, and so you think, okay, 100,000 cells, I've got like, you know, 1 to 5,000 cells active at any given time, 1 to 5,000. I already said it only takes 100 to actually do the accurate localization. Well, what am I doing with the other, you know, 4,900 cells? And so there's this idea that there's additional information that could be encoded that might, you know, that might need to be encoded. And when I kind of get to, we'll take a break, and I get to the temporal coding properties, and that is how these cells not only respond to immediate location, but also past and future location, the so-called phase coding. And you think now sort of the phase coding or sequential dependence. So now you have coding that depends upon the sequence or the trajectory, not just the location. Okay, then now that accuracy, it's not just this location, but I can now think of, you know, what I referred to before, like this context dependency. What is context? It's like anything that changes, anything in the environment that changes that might require that I use that information, map it onto a different response. And so that could be something as simple as, I'm at this location, and sometimes I go left, sometimes I go right? Bifurcating paths. Well, there's the left path, there's the right path. And that's what you see when you actually have animals engage in behaviors in which there might be like a bifurcation in trajectory, different trajectories that cross a single point, you remap, that gets remapped. So now you need to be thinking, oh, this complexity, you know, the dimensionality, 100 might be enough to, you know, estimate current location. But now how many do I need to estimate the potential trajectories that go through that location? And another just tiny little tidbit, what you tend to find is you will increase the diversity, like place field size and other properties, as you increase behavioral complexity. Animals doing more at, you know, one spot will change the nature of the place representations. That could be running, it could be feeding, it could be anything. When you have very consistent boring behavior, doing the same thing, running along, a long corridor, the fields tend to be larger and, sort of less heterogeneous. So representational complexity is tied to behavioral complexity, the need to map it on different responses. So why don't we take a break, and we'll get to the next part, which is just talking about the kind of the dynamic modulation, the temporal coding properties of the hippocampus.